Hello and welcome. Some of you are wondering why somebody without more hair is up here. Filling in for Yaniv, but also happy to be up here in conjunction with our speaker for today. And wanted to briefly explain a little bit about that so everybody knew what was going on. So I'm Alan Love. I'm the director of the Minnesota Center for Philosophy and Science in the Philosophy Department here at the University of Minnesota. And many of you might be wondering why a seminar in plant biology with a well-known plant biologist is being sponsored by the Minnesota Center for Philosophy and Science at the College of Liberal Arts. That doesn't typically happen every day. Vivian Irish is here as a part of a project that I have. It's a 3-year initiative with Bill Wimsatt and other researchers called Integrating Generic and Genetic Explanations of Biological Phenomena, sponsored by the John Templeton Foundation. The goal there is to try and understand from different disciplinary approaches life's complexity, and we have a number of activities ongoing in that regard. So the grant funds, in conjunction with support from CBS, made it possible for Vivian to be here with us today. If you're interested in more details of the project, I'll just direct you to the website, genetic.umn.edu. Now, let me just say a brief word about the Center for Philosophy and Science and why this is the kind of thing that we are interested in supporting. The center is an independent research unit in the College of Liberal Arts that promotes excellence in training in the philosophy of science, but also emphasizes scientific reflection by scientists themselves in order to encourage and better understand the activity of science and why it works and why it might sometimes not work. And so that's part of what we are hoping to do in the endeavor of collaborating alongside of scientists in understanding what is this thing called science. And so if you're interested in some of the activities, a number of you here have participated in some of those in the past. You can check those out at mcps.umn.edu. Now, instead of handing it off to Yaniv, it's my pleasure to introduce today's speaker, Vivian Irish, who comes to us from Yale University, where she's in the Department of Molecular, Cellular, and Developmental Biology, as well as Ecology and Evolutionary Biology. She took her PhD from Harvard. Many of you might not know that she was working on animals. Drosophila was where she began, but then some might say she saw the light and it was Arabidopsis, and she has never looked back. She has worked on a number of topics in Arabidopsis. A lot of what has been intriguing in my own research and thinking about things is the work she's done on petal organogenesis and petal identity. And today, she's going to be talking with us about conservation and innovation in flowering plant morphologies. Please join me in welcoming Vivian Irish.
Thank you, Alan. That was a really kind introduction. Actually, before I start, I need to find a point. Is there a point right here? Yeah. Okay. It's in there. Oh, get a stick. So, yes, I still love Drosophila, I have to say. And I will also say this is not Arabidopsis, for those of you who are not familiar with this. What I want to talk about today is, as the title implies, conservation and innovation. But really, I'm not going to be talking so much about flowering plant morphologies. I'm really going to be talking about conservation and innovation in some of the underlying molecular mechanisms that drive different kinds of floral morphologies. And I'm really going to focus, as Alan alluded to, on petal morphology. So to give you a little bit of background, I understand this is a somewhat diverse audience, and so I'm going to try and cover a little bit of background. Let me start with some basics. I'm sure many of you know the flowering plants, the angiosperms, are a really speciose group. They're very prevalent on our planet. There's over 250,000 or so species of angiosperms, and they have some lovely flowers, as you can see. And as you look across all of these different angiosperm flowers, you can see that there's a lot of diversity in terms of color, in terms of form, in terms of how large the different organs are, in terms of how many organs there are. But there's also a lot of commonalities in these floral forms as you look across them. So let me start with describing to you some of the commonalities, and then we'll talk about some of the differences. Okay, so this is the Arabidopsis flower, much less showy as compared to that water lily flower I had on my first slide. But I want to use this Arabidopsis flower to illustrate the basic body plan of the flower. And actually it was Goethe, the famous poet and botanist, who actually used the term bough plan to explain what these kinds of, the organization of flowers. And so this bough plan or body plan is organized as follows. The flower starts as a young floral meristem, and it starts to elaborate different organs. On the outermost part of this floral meristem, sepals start to arise, and then petals in the next whorl, then stamens, and then carpels in the center. And these organs grow up and mature individually. So the outermost organs you see here are sepals, then the petals, and then the stamens, and then the carpels in the center. The stamens are the male reproductive organs, and the carpels are the female reproductive organs. So this is our general bough plan of sepals, petals, stamens, and carpels. And so as we look across all of those different flowers that I showed you, they all have that same general body plan or bough plan. But there are, of course, lots of different morphologies. So what we really want to talk about or think about is what's conserved and what has diversified across all of these different flowering plant species. Okay, so that's what I've illustrated here. So here's a tree of the angiosperms, somewhat simplified, showing some of the major groups in the angiosperms. And this is in comparison to the other group of seed plants, the gymnosperms. And so I want to point out a couple of things in comparing the angiosperms to the gymnosperms, which are illustrated here by these different pictures. Gymnosperms, let me just show you here. Gymnosperms actually have male cones and female cones on separate axes. So here's a male cone. These are usually lower down in the tree, and here's a female cone. So the cones, the reproductive structures, are on separate axes. And it's not until you get up into the angiosperms that you see all of these different diverse floral structures arise. Hence the name of the angiosperms, which have they have flowers which have an enclosed seed. So at the base of the angiosperms, a number of events occurred during the evolution of this group. There was the enclosed seeds, as I mentioned. There was the origination of the endosperm, along with double fertilization. There was the elaboration of this bisexual axis now, instead of unisexual axis, as you see in gymnosperms, to have a single axis with both male and female reproductive structures on it, as illustrated up here. And the other thing that occurred at the base of the angiosperms was the elaboration of what we call perianth, or these sterile organs that surround the reproductive organs. What's illustrated here is the sepals and petals surrounding the reproductive organs. So all of these changes that occurred at the base of the angiosperms is what Darwin alluded to as an abominable mystery. And this is still an abominable mystery. We still don't know, and I'll tell you right now, we really don't understand how all of these characters came to arise and contributed to this enormous diversity of angiosperm on our planet today. That being said, we know a little bit about some steps. And so what I want to focus on today is to talk about the elaboration of petals as part of this explosion of the angiosperms. So what are petals? Obviously, you probably think you know what a petal is, but I will tell you what my definition of a petal is. And I define it on the basis of two characteristics. One is its position. So petals are always immediately surrounding the reproductive organs. And petals generally are these large, showy structures. So they have a particular morphology. They're often characterized by these very specialized cells, which are actually give the petal a sheen. So these cells are actually really cool. They act like CDs. You know how CDs have a sort of a sheen to them? These little striations on these cells give petals a sheen as well. And so petals are characterized by their position, by their morphology, and they're also thought to have evolved to increase fitness through facilitating pollinator attraction. Although there's some controversy about this because the insects evolved probably well before angiosperms did. But still, there probably was some coevolution between insects and flowers. Okay, so we've got a definition of petals, but it turns out there's actually a lot more variation as we look across the angiosperms. So let me just describe to you sort of some general character, some general classes of petals that we have. We have a bipartite perianth in species like Arabidopsis, where we have distinct petals and sepals. We have situations like in this tulip tree flower, where we have the perianth organs all looking very similar to each other. And so these are often called tepals. We have situations in which petals have been secondarily lost. And then, for instance, in the grasses, there can be other types of organs surrounding the reproductive structure. So in grasses, for instance, there's a specialized organ called the loticule that sits in that position where petals normally appear. Okay, so we have this diversity of petal types, and as we map this diversity out across the angiosperms, what you can see here is that across the angiosperms, there really is a lot of diversity in whether or not they have distinct perianth structures, distinct petals, where they lack petals, or they have a mixture of different characters. And so we can just mark these situations in which we have bona fide petals in the bipartite perianth here in these pink highlighted areas here. So petals have arisen probably multiple times during the evolution of the angiosperms. Maybe didn't arise as petal organs per se at the base of the angiosperms, but have elaborated multiple times. Or perhaps petals originated at the base of the angiosperms and have been lost multiple times. So to really start to address this kind of issue, what we want to do is ask at the molecular level, to what extent do all these petals and petaloid organs use the same kind of developmental program? And then the second question I want to get to is, as these morphologies change, how do these underlying programs change? So I'm going to tell you one story first, to really start to focus on some of the conservation and diversification and the underlying molecular programs that drive petal specification. Okay, so to do that, I need to tell you about a couple of genes that we've been working on for many years. The plant biologists in the audience will be very familiar with this, I'm sure. These are the so-called V group of Matchbox genes. And again, I'm going to use Arabidopsis as my reference to explain this. So in Arabidopsis, there are two such Matchbox genes, A-petal-3 and pistillata, abbreviated AP3 and PI. And these two genes are both necessary and sufficient to specify petal and stamen identities in Arabidopsis. So we know that because loss of function mutations in either A-petal-3 or pistillata result in the elimination of petals, as well as the elimination of stamens, and result in homeotic transformations of those organs. And if we ectopically express both AP3 and PI outside of their normal domains, we get conversion of, for instance, the first world organs, which would normally form sepals, into petals. So we get ectopic petal and ectopic stamen identity. So AP3 and PI are necessary and sufficient for specifying petal and stamen identity. They're both encode Matchbox containing transcription factors, which have a highly conserved DNA binding domain followed by several other motifs. And a motif I'm going to mention again later on in the talk is this K domain, which is important for dimerization and protein-protein interactions. And these transcription factors, like many transcription factors, act as parts of larger transcriptional complexes. So I'll tell you just a little bit about some of the biochemistry of these two Matchbox gene protein products. So the products of these two genes, AP3 and PI, encode transcription factors, and as transcription factors, they should bind to DNA. But in fact, purified AP3 protein or purified pistillata protein cannot bind to canonical target sequences. They only bind to DNA as an obligate heterodimer. So you need a dimer of pistillata and AP3 to bind to DNA. Okay, and the kinds of DNA they bind to a number of different target sites, but it turns out that one target site of this AP3-PI heterodimer is the AP3 promoter itself, which you see here in this chromatin immunoprecipitation assay. So AP3 and PI proteins bind as heterodimers, and they auto-regulate the expression of AP3. And it turns out that there's actually auto-regulation of both AP3 and PI. So here we have a little cartoon of AP3 expression very early in flower development and then just slightly later in flower development. And this is this domain that's going to give rise to petals and stamens, if you recall from the earlier cartoons. Pistillate expression is a little bit more broad, but then comes to be resolved to the same domain as AP3. You can see this here in this immunostain, the AP3 protein and PI protein. So the way these gene products get localized in this region is through autoregulatory feedback control. So we can draw this as a circuit diagram, which looks a little bit complicated, but it's pretty straightforward. The AP3 gene produces its protein. Pistillate gene produces its protein. They dimerize. They autoregulate AP3. They positively regulate its expression through binding directly to the AP3 promoter. And they also positively regulate the expression of PI, although this appears to be indirect, so through several other intermediary steps. And we know this circuit is important for petal development. Okay, so I've sort of quickly gone through the biochemistry, but I want you to sort of understand what these proteins are doing, because that will be important as we go on. Okay, so just to summarize what we know in Arabidopsis, these two Matchbox gene products act as a heterodimer. They function to specify petal and stamen identities, and they participate in these auto and cross-regulatory events to maintain this expression domain, this B domain of expression that is the region that's going to give rise to petals and stamens. Okay, so that's what we know in Arabidopsis. But I want to give you the punchline of the first part of my talk right here, so I can sort of walk you through the reasoning as to how we get this. I want to demonstrate for you that the situation of Arabidopsis is actually fairly unusual. It represents probably a secondarily derived and certainly streamlined version of this regulatory circuit. And what I want to talk about for the next few minutes is how this network is actually diversified in different ways, in different lineages. But as a whole, this network has maintained its function. Okay? So let me introduce to you how we actually came to that insight. So over the years, we have been looking at the diversification of the AP3 lineage genes and the pistillate lineage genes across the angiosperms. And other folks have been looking at it in the gymnosperms as well. And I'm really cutting to the chase here and just showing you a little bit of that ton of data to illustrate a few salient points. Okay, so here is a very, very, very simplified tree of the angiosperms with just a few taxa picked out for reference, and here's our sister group, the gymnosperms. So prior to the split of the angiosperms and the gymnosperms, through our reconstructions, we think there was an ancestral form of an AP3PI hybrid gene that persists in the gymnosperms. In the angiosperms, there was a gene duplication event that probably predated the diversification of the angiosperms. And then there were many subsequent gene duplication events. And the way these gene duplication events are labeled is that all the AP3 homologs are diagrammed in blues and greens, whereas all the pistillate lineage genes are diagrammed in reds and yellows. So we have an ancestral AP3PI-like gene that's duplicated, and then we have various other duplication events still. And so you can already see here, Arabidopsis has actually lost some of these ancestral gene copies and now only has one AP3 gene and one PI gene. What we decided to do was to look in a different clade to try and address what these gene products were doing. And we decided to tackle this question in the Solanaceae. So the Solanaceae, this family includes species that I'm sure you're familiar with. It includes Petunia, Petunia hybrida. It includes tomato. And it includes Nicotiana benthamiana, which is actually a diploid precursor to cultivated tobacco. So we decided to work on these three species to try and parse out what's going on in a situation where we have more complex numbers of AP3 and PI genes, and try and understand the diversification of this regulatory network. So this illustrates what's going on here in a little bit more detail. So the Solanaceae probably diverged about 30 million years ago or so. Prior to that diversification, we had two AP3 lineage genes and two PI lineage genes. I've color-coded everything, so the names are confusing, so hopefully you can follow everything just by color coding. And these four persist in tomato, nicotiana, and petunia. Okay? So we have orthologs of all these genes in all the species that we're looking at. So we decided to start doing some functional studies to try and get at what was going on in terms of what these gene products are doing. So let's start with tomato. So here's our tomato flower for reference. If we just look at the expression patterns, and this is just a snapshot of one stage of expression for the two AP3-like genes and the two pistillata-like genes, you can see that they're expressed in the flower primordia. They're expressed in slightly different domains. So for instance, if we look at the two AP3 genes, one AP3 gene is expressed predominantly in this B domain, this domain that's going to give rise to petals and stamens, and this other AP3 gene is expressed orthologically. There's also some differences in how the pistillata-related genes are expressed. Okay, so there's differences in expression of these genes. So we decided to ask, what are the differences in functions of these different gene products? And so what we did was we identified a mutation in the tomato AP3 gene, and we used an RNAi approach to knock out the function of the other AP3-like gene called TM6 in tomato. And I think you can see just from the flowers here that they have produced very different phenotypes. So here we have an AP3-like gene. When we eliminate its function, it shows the canonical homeotic transformations of black petals, where in this case the petals are perfectly uniform. And in fact, it's just the stamens that are somewhat defective, as you can see here. Okay, so the two AP3 genes have two different types of functions. So we wanted to ask, was this difference in function due to differences in those expression patterns I showed you, or due to differences in what the proteins were encoding? So what we did was a rescue experiment. So we took our AP3 loop line, and we reintroduced either the TM6 gene or the AP3 gene back into that mutant background. And I know this picture is probably not so clear for all of you to tell, but we always got a greater degree of rescue when we reintroduced AP3 into the AP3 mutant background. And we got a much less good rescue when we reintroduced TM6 into the same background. So what that argues is that these genes are not only expressed differently, but the protein products have somewhat different functions as well, because we never get good rescue when we're expressing it in the exact same way as we express the other. Okay, so we started to parse out what's going on with the tomato AP3 genes, and I'm just going to now sort of summarize a lot of work over a number of years to try and compare what these genes are doing. So using the same kinds of approaches I just described to you, we started knocking out the AP3 copies of these genes and the PI copies of these genes in tomato, tobacco, and petunia. So let me explain to you what these cartoons mean. So where we have a cartoon that's all white, that means that's a normal situation. So of course in normal wild type tomato flower, we have normal organs in the Christiana flower, and normal organs in the petunia flower. If we knock out all AP3 PI function, complete loss of that gene circuit, we get defects in petal development and defects in stamen development. So that's cartooned here in green. So in each case, the B group gene function as a whole is required to specify petals and stamens. But if we look at the function of each gene individually, we see there's a lot of variation. So I already sort of walked you through the comparison of the AP3 and TM6 mutations in tomato. But as we look across the orthologs, for instance, in the different species, or we look across the paralogs within a species, we can see there's a lot of difference in terms of their functionalities. So, for instance, the AP3 gene in tomato is required for both petal and stamen development, it's required for both petal and stamen development in tobacco, but it's only required for petal development in petunia. So we can look at the loss of function situation and infer what the normal activities of these gene products should be, and they're different in all cases. But if we eliminate, as I said before, if we eliminate all B group gene function, we see the same phenotype across all of the species. So the individual components are acting differently, but the circuit as a whole is acting similarly. Okay, so we went on to ask these proteins were doing in terms of how they were heterodimerized. So if you recall, I told you when I described you the rhabdosis situation, there were rhabdosis AP3 and PI proteins heterodimerized. And so we compared the heterodimerization capabilities of the AP3 and PI genes from tomato, nicotia, and petunia, and this is what we found. So in tomato, we found we could get heterodimers of one AP3 and one pistillata copy, and one AP3, the other AP3 copy, the other pistillata product. But we saw more diversity in tobacco and petunia. So there was differential partner preference as well. Okay, so these gene products have different functions individually, but the circuit as a whole is maintained. And they can form different heterodimer combinations as well. Okay, so we went on to ask whether or not the maintenance of these heterodimers is being driven by positive selection. So the idea here is that these heterodimers are required for specifying some aspect of petal and stain individual. And one might naively think that if you establish such a function that is critical to specifying these organ types, it would be very stable and not be subject to positive selection. So we wanted to look at that, and so what we did was we looked to see whether or not there was a greater amount of synonymous changes versus non-synonymous changes, or non-synonymous changes versus synonymous changes, to determine whether or not there was positive selection at any of these genes. So we characterized this, and we found that there was positive selection occurring specifically in the K domains of several of these genes. Okay? Like one AP3 gene and one pistillata lineage gene in each case here. So these circuits are evolving, but they're maintaining a particular function, but that the individual components are rapidly evolving and under positive selection. Okay, so let me summarize this first part. So in the Solanaceae, we see diversification of individual gene functions, but the combined action of these gene networks is conserved. And we think this can be explained in part by this requirement for heterodimer function, involving members of each of these two lineages, the AP3 lineage and the PI lineage. But these protein complexes are being adaptively maintained because we find evidence of positive selection, specifically in the regions that are required for heterodimerization. And furthermore, just as an aside really, it appears to be the more derived genes that appear to predominate in petal function, specifying petal development. Okay, and then the last thing I want to mention about this is, as you may have seen, tomato actually had fewer heterodimers being formed as compared to tobacco or petunia. And tomato actually seems to be losing some of these interactions. So the question is tomato actually going the way of rhabdosis, streamlining its circuit that hasn't quite gotten to the point of losing all of those components yet? It's an open question. Okay, so I want to actually shift a little bit from our work on the Solanaceae and tell you about another comparison we did, looking in the pottings. And so let me tell you why we did this. So I told you now, rhabdosis is a simple streamlined circuit of one AP3 lineage gene and one PI lineage gene that work together as a heterodimer to specify petal and stamen identity. There's been gene duplication tests in the lineage leading to the Solanaceae, where we have 4 genes now that each have acquired different capabilities, but the circuit as a whole is maintained. So we wanted to do a comparison and ask, is this kind of situation holding in other lineages of the Megastory? So we decided to work with the power somniferum, which has, again, gene duplicates, but these duplications have arisen separately from the gene duplications we see in the Solanaceae. Okay, so we had to develop the power somniferum as a model system, so that took some work, but we got there in the end. So here are some in situ hybridizations to show you the expression patterns of these 2 AP3 lineage genes and these 2 PI lineage genes. And again, you can see, although their expression patterns are similar, there are some differences as well. So for instance, in this case, the AP3-1 gene in poppy is expressed in petal, whereas the 83.2 gene is not expressed in petals. And I'm not going to go through all the poppy work, but we basically did the same kinds of analysis in poppies, using virus-induced gene silencing to knock out individual copies of each gene and knocking out the circuit as a whole. And if we knock out the circuit as a whole, we get this very dramatic phenotype. So this is a wild-type poppy flower just as it's opening, and this is our virus-induced gene silence knockout of that entire circuit. And you can see, again, we lack petals and we lack stamens in this homeotically transformed poppy situation. Okay, so let's go back to sort of our overview here. We also looked at heterodimer formation in poppies, and in this case, we found that we could get homodimer formation as well in the poppies. So the way these proteins dimerize is distinctly different in poppies as compared to Arabidopsis or the Solanaceae. So we think that there's compensatory variation in how these protein complex components are acting in terms of maintaining this overall function. And that the robustness of this network, somewhat surprisingly, I think, depends on positive selection. And furthermore, those heterodimers that are specifically involved in petal development depends on different subsets of these B3 genes. So in Arabidopsis, we only have one heterodimer that's involved in both petal and stamen development. In tomato, it's only one that's involved in petal development. All of these in Nicotiana and Petunia that are involved in petal development. And in Papaver, it's only this one that's involved in petal development. Okay, so to summarize this first part, we think that the ancestral role of these genes was in male reproductive development. And somewhere early in angiosperm evolution, these genes were recruited to specifying petal development. How that initially occurred, we really don't understand that. But in these different lineages, the specific modes by which these genes are acting now is diversified considerably. Nonetheless, the overall circuit has been maintained. So the alteration in function is contributing to this diversification, but at the same time, we're retaining this, what I'll call a developmental module. And I want to call this a deep homology, that there's a deep homology at the level of this circuit. And just as a question, we really don't know the answer to this. Is positive selection driving the maintenance of function of a circuit and overall a general theme in evolution? I don't know, but I think it's a tantalizing possibility. Okay, so I'm going to move on to, oops. Move on to, oh dear, it's always something, right? Give you guys a little breather while I find my place. Okay, so we've talked about how petal identity is established in these different examples that I talked about in the Papaveraceae, looking specifically at Papaver somniferum and in the Solanaceae and also in Arabidopsis. What I want to do now is switch gears a little bit and talk about a somewhat different aspect of petal development and talk a little bit about how the pathways that produce petals may have been modulated to produce petals of different sizes, shapes, and colors. And in particular, I'm going to talk about different sizes of petals. So to do that, again, I need to go back to Arabidopsis and just remind you that we actually do know a fair bit about the gene circuits that are involved in driving petal development in Arabidopsis. So what I've illustrated here is sort of a network diagram or a circuit diagram of the genes for which we have functional characterization of how these gene products operate to regulate each other. You can see it's a very complex circuit. You now know part of this circuit is APTPI circuit. And together these genes are all involved in different aspects of petal development. So I want to tell you about a different aspect of this circuit. And I'm going to be focusing on some more recent work we've done to look at this gene called rabbit ears here, which is expressed specifically in petals and is involved specifically in petal development, how it regulates these three genes, these three TCP genes. Okay? So first I'm going to tell you about our work at Arabidopsis, and then again extend that to some other situations. Okay, so to do that though, let me tell you a little bit more about how petals grow. So plant development is actually very easy to understand in that there's no cell movement. So where the cell is positioned is where it stays. So it's really easy to illustrate how an organ like a petal grows. So petals grow by an initial phase of cell divisions, which you can see here in this line where all the mitosis are being marked by this labile marker here. And so we'll mark that as green. You see lots of cell divisions. As the petal grows, you see cell divisions predominantly in what's called the blade. Then the cell divisions start to decrease from the tip to the base. We maintain cell divisions towards the center. And eventually, cell divisions cease. And the final phase of petal development, and this is in general across all the angiosperms, has to do with postmitotic cell expansion to result in a petal, like a claw. Okay, so this is how the petal grows. So rabbit ears, as I briefly mentioned, is involved in specifying petal development specifically. And it's expressed just in the domain that's going to give rise to petals. Now, we know rabbit ears is important for petal development because in the mutant situation we get abnormally small petals, which you can see there's one here and one here. And this appears to be due to a reduction in cell divisions. Okay, so we can see that graphed out here. Here's our wild type situation here. And in our rabbit ears mutant, we see a reduction in the numbers of cell divisions. Okay, so rabbit ears is required for promoting cell division in the developing petal. So, Tango Wang, a postdoc in the lab who was working on this, did an RNA-seq experiment to identify targets of the rabbit ears gene, and I won't go through that analysis, but he identified 3 TCP genes as targets of rabbit ears, and we were very excited about this. So let me tell you why we were excited about this. So the TCP genes encode a different class of transcription factors. They encode a basic helix loop helix group of transcription factors, and Arabidopsis has 21 TCP genes in its genome. And many of these genes have been implicated in growth control in other contexts. So we thought these were great candidates for being involved in growth control in the petal. And it turns out that rabbit ears negatively regulate 3 of these TCP genes that have really not been very well characterized at that point. So you can see that rabbit ears negatively regulates TCP5, TCP13, and TCP17 because if we look at the expression of these 3 genes in a rabbit ears mutant as compared to wild type, we see the expression go up. And if we transiently overexpress rabbit ears, expression goes down. So that's a good hallmark that rabbit ears is regulating these 3 TCP genes. So it turns out nicely that these TCP genes are expressed in petals, but they're not expressed at early stages of petal development. They're expressed at later stages of petal development in somewhat overlapping patterns. Okay? So they're good candidates for being involved in petal growth control. So we wanted to actually show functionally that they were involved in petal growth control. And it turns out that single loss of function mutants really don't show any differences compared to wild type. But if we knock out all 3 of those TCP genes that I mentioned, TCP5, 13, and 17, using an artificial microRNA, we can see that the petals are enlarged. Okay? And in particular, it's the petal width that's enlarged. And that results in an altered shape of the petals. And furthermore, if we overexpress one of these TCP genes, for instance, TCP5, we can recapitulate a sort of a rabbit ears type of phenotype where we get small petals. Okay, so these TCP genes actually do regulate cell divisions in the petal, and we know this by actually counting cell again, using the reporter I showed you before, here's a wild type panel of development of the petal. Here's the knockout, the triple knockout, and we see, maybe hard to see in the back, but we still see cell divisions even at this very late stage. See blue dots here. And if we overexpress one or more of these TCP genes, we see a reduction in cell divisions and a reduction in the petal size. And it turns out that this change in cell divisions is really a change in the duration of cell divisions in the petal. And we can quantitate out that. So these TCP genes are basically acting as brakes on cell division. If we overexpress them, we get a break in cell division early. If we lose their function, we retain cell divisions later in development. Okay, and so we also showed that TCP5 in particular largely mediates this rapid Weir's function in petal growth. And we can do that by introducing a TCP5 mutant into a rabbit-ears mutant background, and we can largely rescue the mutant phenotype. So this suggests that rabbit-ears indeed is negatively regulating these TCP genes, in particular TCP5, and in turn, these TCP genes are acting as a brake on cell division. Okay, so early in petal development, rabbit-ears is on, as I've shown you. And so it's repressing this repressor of cell division, so this pathway is off. Rabbit-ears goes off later in petal development, so that alleviates this repression, turning these genes on. These genes then can act as a brake to cell division. You get the terminal stages of petal differentiation as a consequence. Okay, so I told you a lot about these TCP genes and why we were interested in them. We've done a lot of work in Arabidopsis to try and understand their roles in petal development in Arabidopsis. And I'm going to sort of segue into something different and then come back to the TCP gene. So, and hopefully you'll see what the tie is. Okay, so as we look across different Arabidopsis ecotypes or strains of Arabidopsis, there actually is a lot of variation in petal size. Okay, so here we're looking at just a few different wild type lines here, and you can see even just across different ecotypes, there's quite a lot of variation in petal size. And so we wanted to take advantage of this diversity of petal sizes to just do a non-specific screen to try and identify the genes that were involved in regulating petal size. So we undertook a QTL analysis to try and get at these genes, and we used two different populations to try and get at this. We used two different recurrent populations. One is a cross between the two strains, Landsberg recta and Columbia O. The other is a population generated between Estland and Columbia. And these are populations generated by Lister and Bean in the first case, and Dallas-Brown and Delafido in the second case here. And so the idea here is we can use these recombinant inbred lines to identify quantitative trait loci that are influencing different aspects of petal growth. So we scored 20 petals and 20 sepals as a control in 100 recombinant inbred lines from each of these populations. And we scored these petals for length, width, shape, and we normalized the cells and sepals. And then we calculated the positions of the resulting quantitative trait loci. I'm not really going into the details of this, but basically we can use this to map variation in the genome. And so this summarizes that analysis. And really, so there's a lot of data presented on this slide here, and I want to walk you through a few different points that I want to make here. So first of all, these are the 5 chromosomes of Arabidopsis, showing where all the different QTLs that we identified are located. And the QTLs we identified from the Columbia across to Landsberg in blue, and from Columbia across to Estland in pink. And the first point I want to make is that a lot of the QTLs we identified in one population were distinct from the QTLs we identified in the other population, suggesting there's a lot more allelic diversity out there that we haven't even begun to capture. The other point I want to make is that on this panel, I've marked many of the genes that we know of from genetic analysis that we know have something to do with petal development. And I think you'll see that in most cases, these QTLs don't map on top of any of these genes that we know of from genetic analysis that are important for petal development. With the the exception of one, and this is where I'm going to bring it back to the TCPs. This one particular QTL maps right on top of TCP5. So one of the things that we're currently interested in is trying to determine if variation in the TCP5 gene, which I've already shown you, is important for regulating petal growth by regulating the duration of cell divisions. If this particular QTL is responsible, that the TCP5 is responsible, because the gene underlying this particular quantitative trait. Okay, so I've sort of run you through a lot of different things. So let me sort of summarize the broad themes that I tried to hit on through today's talk. So first of all, as we look across the angiosperms, petals as we define them on the basis of position represent a deep homology amongst the angiosperms. And the origin of petals is correlated with this activity of these B group genes. And it's this circuit we think is really the deep homology. The circuit action is really the deep homology that's driving petal specification in all of these different lineages. But how the individual components of this circuit are deployed, how these individual group genes are deployed in these different lineages differ. And so independently derived petals may actually represent different combination of B group gene activities. And so we think that this might represent a case of parallelism. And so in the last part, what I tried to convince you of is we're starting to get a handle on some of the genes that we think might be important for some of the downstream components of petal specification. And so some of these genes may be important for species-specific differences in the sequences in petal size or petal shape, etc. And we think that TCP5 might be at least one of such genes. Okay, so I'm going to wrap it up by reminding you, although I probably don't need to remind this audience, that understanding the evolutionary history of genes in plant development is really important as we face a crisis with climate change and a burgeoning population. We're going to have to feed these people. And so while we're not explicitly working on trying to feed the world, I think understanding how these regulatory circuits operate and how they've diversified over time is going to be really important as we think about engineering new crops for the future. So I'll leave it there, and I will thank the people who are involved in the work. I've tried to review a lot of work from my lab over a number of years, and so all the people that contributed to the projects I've talked about are listed here. We're currently still continuing our Viva work. Faye and Pascal are actually working on a project in citrus, which is a new project, to try and apply some of what we're learning to a more relevant crop species. And I also want to thank many collaborators over the years who have helped us with all of our Viva work. And also, in particular, mention the NSF, which has been instrumental in funding all of our Viva work over the years. So stop there, and I'll take any questions. There's a lot of variability in it. We haven't even begun to address it. Yeah, so we're just really scratching the tip of the iceberg.
Are you going to look at other populations?
No, what we try to do is we try to actually define what genes we can have been already identified in these QTLs. And actually, we've identified one, which is a receptor-like kinase. It's sort of a different story. Yeah.
So for AP3 group, is there any alternative splicing in tobacco and petunia?
We didn't find any signs of alternative splicing.
Oh.
No. Yeah.
Can you talk about this tension between positive selection and stability? It seems, or robustness, it seems like positive selection makes you go in ways that, it seems like you probably have some thoughts there, but I couldn't figure out exactly what they are.
I think it is a tension, and it's something that I think about, every day I think about it differently, I guess is how I would put it. I mean, I guess the sort of, the general bottom line that I don't think anybody here would quibble with is that evolution is not static. Things are changing all the time, right? And so basically, I think the robustness of the circuit is really, really important for maintaining this organ identity. And so whatever is happening at the gene level is just sort of pulled into place. And so positive selection is constantly maintaining that, is driving that circuit, but then we're maintaining the heterodimer formation by requiring that there's positive selection on both halves of that heterodimer. Maybe I didn't make that clear. Okay? But that circuit as a whole is robust. I'm not sure I'm answering your question. This probably is like an over-beer type of question.
But also the function of the petals is primarily to attract pollinators.
Yeah.
You have an awful lot of degrees of freedom when you do that.
I know, so you wouldn't think that necessarily it would be such a robust circuit for specifying petals, but it seems to be.
Yeah.
I agree. Yeah.
So I have two questions. So one question is related to Yanni's question.
Yeah.
So you saw the QTL with the kind of, pedal morphology. So each of them, are they really defining the different, global states, or it's more of the just dialing things a little different?
I didn't get the end of that, sorry.
And let's put it in this way. So how much of the pedal morphology difference you see with a single accession and then just different environment?
How much variability do you see? So pedal variability is actually very little. So a single accession, so that was actually something really important for the QTL analysis. Pedal variability within an accession, within a string, across different environments is very stable. So that actually made it good for doing QTLs. What was the, I think you had a first part of your
So,
question.
I was asking whether those QTLs are really defining the different robust states of the network, or so just dialing a little bit differently, say, some environmental or the hormonal things, so just different sensitivity to it.
Yeah, I would probably argue against the environmental effect because we really looked at that carefully.
And the second,
Oh, sorry.
sorry, one more question is so you assume that the common ancestor of gymnosperm and angiosperm didn't have the AP3PI separation.
Right.
So why do you think that heterodimer requirement evolved?
Good question. I don't know is the answer, but it did evolve, and it seems to be an absolute requirement in this circuit. I mean, you start with homodimers, it's clear. So heterodimerization evolved from homodimerization. So if you go back to the basal lineages of the angiosperms, you see, and you could even see this in the poppies, you see cases where you see homodimerization. But in the core eudicots, for instance, you never see homodimerization. So you've lost that ability. So maybe concomitant with that loss of homodimerization, you now, when you have to sort of enforce heterodimerization, you then get this positive selection on both halves. You have to maintain that circuit that way because you've lost the capability to homodimerize. Just BS. Yeah, you got it.
So you're mentioning the potential that one of your QTL coming from S1 was TCP5. And I know you still have work to do to prove that, but let's just assume it is for a second. If you look at the sequence, can you guess whether it's due to a difference in the ability for rabbit to regulate that allele of TCP5? Or is there a functional difference? Is that protein coding sequence different?
We've looked at we don't see any differences in the protein. So we've done the easy bit, which is to look at the protein coding sequence. And we don't see any allelic differences in these different strains. So we think it is a regulatory difference. That being said, it's hard to see that by expression differences. I mean, because we're looking at something quite subtle. It's only explaining 10 or 11% of the difference. So I don't think we can see it by expression. So then we're going to have to go in and do some more sophisticated tests. So that's where we're at, yeah.
So I was wondering about some of the deepness, whether it's really due to the fact that most of the downstream genes act throughout the plant. I mean, it's like your TCP shape and,
Mhm. So, yeah.
so I mean, you've got these two genes that are
Yeah.
specialized to basically control genes that act throughout the whole life
Yeah.
cycle of the plant,
Yeah.
specifically in petals, which kind of drives that.
Yeah, so I mean, I think a lot of what we're gonna see as we look at specification of particular organ types, let's say, which is what we're looking at, is, yeah, you're just modulating the expression of basically housekeeping genes from cell division, cell cycle, synchronization, et cetera. All those different housekeeping functions, but they're under, they have to be deployed in just such a way,
Right.
right? So,
And not messed up in other places.
yeah.
I want to ask you a Drosophila question.
Okay. Oh, boy, I have to think back.
Your petals, and you're staining them for proliferation, they look a lot like wing discs.
Uh-huh.
And so some of your TCP knockdowns, they're more wide than they are larger.
Uh-huh.
Do you ever see proliferation pattern in such a way that explains the widened petals, something like a DTP Brinker axis in flies?
No, so the answer is no, and it's probably the time, well, we think it's the timing of these TCP genes that's spreading. So as they turn off, then we see, I'm not explaining through, you're not getting tons and tons of mitoses, you're getting sort of patches of mitosis, or little spots of mitoses throughout the petal, right? It's not every cell is divided.
Sure. And how do you get the widening?
Oh, so yeah, we do think that there is a retention of mitoses across that band in the middle. Is that what you're asking?
Yeah, I'm asking
Yeah.
if it's regulating
Yeah.
shape more so than
Yeah.
size, and if you have any handle on how it's doing that spatially.
Yeah, so what we need to do, and we haven't really gone back and done this very carefully, but it's on the list of things to do, is actually look at mitoses across the width. We have to sort of survey top to bottom before, and we need to look more carefully across. But yeah, that's good.
This is really an ignorant question, but I know nothing about it. Isn't olfaction important also in tracking pollen?
Scent.
Pardon?
Scent? You mean? Olfact.
Yeah.
Yeah. The insects smell and the plants produce volatiles, yeah.
Right.
So the petals do produce volatiles.
So the petals do that also?
It's also, it's generally the petals in most species that produce the volatiles, but there are other tissues that can produce volatiles as well, that are in the flower.
Are there localized cells that you can see that are releasing the scents? I mean,
I don't
what
think
I'm
so.
sort of wondering
I mean,
is
it's-
whether there's different components for, say, visual and there must be.
Oh, yeah, I'm sure there is, I'm sure there is.
But whether they co-vary in any interesting ways, or whether you have, I assume these things you've been looking at are connected with optical.
Yeah,
But maybe that was just ignorance on my part.
Yeah, I mean, yeah.
So would there be more things to look at for?
Yeah, so Arabidopsis doesn't really produce pollen, but Arabidopsis, first of all, is self-fertilizing, so it's not attracting pollen. It doesn't have any of these volatiles that roses have.
So it's a single organism for that.
Well, yeah, so it's not really a good model for that.
Yeah.
So people are working on other systems, so roses actually, that kind of, we haven't delved into that at all. I'll just leave it at that.
Okay.
Okay, join with me in thanking Vivian.